Everybody for coming. And I don't know how many of you may have seen the first one of these that I did a little over a year ago, but I hope this one will be better because I have a better internet connection. So presumably I won't sound like I've been drinking all day. And the connection speed will stay constant throughout. So let's hope for the best. You may not know it, doesn't look like it, but I'm in the Valdosta Public Library right now. And they apparently have good internet, so I guess we'll find out over the next few minutes or so. What I'm going to talk about, as you can see, is some of the aspects of reproduction in long-nosed armadillos. Long-nosed armadillos, for those of you who may not know, are in a single genus called Dasypus. And I think you can see from the pictures why they're called long-nosed armadillos. They all have this long snout that they use in foraging for their invertebrate prey in the soil. Now, to begin, I'm going to talk about a couple of, or one thing sort of tangential to the main topic of the webinar, which is, why did I come up with this title? Whoops, where did it go? Sorry. Oh, I'm sorry. I don't know. You may be able to see that. I can't see it because. But why the title? I think anybody who knows who I am knows that my wife Colleen and I have spent a lot of time studying the 9-banded armadillo, Dasypus novum cinctus, which has this really large geographic range that extends from the US down into northern Argentina. But as Anderson is well aware, and apparently a lot of other people are as well, it's a very poorly kept secret. But much like the silky anteater, which has a large distribution, largely in South America, but turns out that within that large distribution, there were actually a number of different distinct species. There is no 9-banded armadillo anymore. And this distribution is not the distribution of a single species. This is preliminary. Don't cite this, but I'm just giving you a heads up, a warning of what's coming. So you need to get used to it. But according to the work that Fred Dilsuck and his grad student, Mathilda Barté, have done, there are at least 4 species instead of 1 for Dasypus novum cinctus. So the actual Dasypus novum cinctus is found here in South America, east of the Andes. There's another species found in the Guyanan Shield. There is a 3rd species found here on the west side of the Andes up into Central America. And then there is a 4th and final species found actually from Costa Rica up through Mexico and into the United States. And that is Dasypus mexicanus. So this work is in review. So as I said, please don't cite it. It should be out. We're all hopeful the paper will be accepted pretty soon, and we're going to have to learn the new terminology. But again, as a lot of you know, Colleen and I wrote a book called The 9-Banded Armadillo. If you have a copy, or do a little gratuitous plug here, the University of Oklahoma Press is issuing a paperback version of this in the spring of this year. So if you have a copy, or if you're going to buy a copy, get it out and do this, because it's not the 9-banded armadillo anymore. Get rid of it. It's the Mexican long-nosed armadillo. Get used to it. And we're trying to get used to it. Taken me a while, but this is one of the reasons for the title, because I figured if I provided the title that said the odd reproduction of the Mexican long-nosed armadillo, nobody would know what the hell I'm talking about. So I decided to just say long-nosed armadillos because presumably, at least generally speaking, a lot of what I want to talk about probably applies to all of the long-nosed armadillos, not just the Mexican long-nosed armadillo. And so just be aware, there are additional species that we're going to need to recognize. And so we'll see how that all plays out. So to get to the bulk of the talk here, or the webinar, I wanted to give you first off an overview, to the extent that we can, about the reproductive phenology of long-nosed armadillos. Now, some species are not well known, so I'm not sure that this necessarily applies precisely to every species, but I think generally speaking, the basic features are probably relatively the same. So phenology concerns what happens over the course of the year. So if we start in the summer, long-nosed armadillos breed in the summer. So that's when mating occurs. So up here in the United States, they would be breeding in early summer, say June, maybe early July. I guess for some of you in South America, that would be going on right now. It probably even started maybe in December, but breeding would be occurring in the early summer. And so that's when females are inseminated. Now, even though they've been inseminated in the early summer, then nothing happens for an extended period of time. So the female's egg has been fertilized, but it's not until the late fall that anything more happens, because there is this period of delayed implantation, also referred to as diapause, where you can see the real thing down here below, but this is an illustration of the female uterus. This would be an ovary here. Here's the other ovary. This little area here is called the fundus, and that would be the same thing that you see right here. This is the fundus. Here's an ovary. There's an ovary. And the fertilized egg travels to the fundus, where it just sits. And it doesn't implant. Embryonic development doesn't begin. It just sits there. And it sits there for a variable length of time, typically until sometime in late fall or early winter. But from early summer until late fall, even though fertilization has occurred, nothing's happening. Now, once you get to late fall or early winter, so again, in the US, this would be about December or so, November, December. For those of you in South America, this would be more like May or June, I suppose. But that's when implantation finally does occur, which means that embryonic development begins. And that's where something really unusual happens, which is that once the fertilized egg implants, you get this phenomenon called polyembryony. Polyembryony just means that you're producing multiple embryos from a single fertilized egg. So what happens is that once the fertilized egg implants, it divides. So it splits in two. And in some cases, those two may split again, and they may split even after that as well. In the case of the Mexican long-nosed armadillo, see, I'm learning, I'm using that name. You wind up getting 4 embryos. So these are embryos of the Mexican long-nosed armadillo here. You can see down here, this is actually, you can't see who this is, but this is Augustine Abba with a handful of what used to be Dasypus hybridus juveniles. So you can see that the number of embryos that are produced is variable, and I'll have a little bit more to say about that later on. But this is really unique. It's only the long-nosed armadillos in the genus Dasypus that routinely reproduce via polyembryony, where they always produce multiple embryos from a single fertilized egg. And so what that means, and I'll come back to this as well a little bit later on, is that all of these juveniles are genetically identical to one another, because they're all derived from a single fertilized egg. So when that splits, all the genetic information is shared between each of the resulting cells that have been created. So there's no other vertebrate that does this. It happens occasionally. In humans, we get monozygotic twins, for example, but that's a rare event. Long-nosed armadillos are the only vertebrates that we know of that reproduce in this way. There are some insects that do something similar, but as far as vertebrates are concerned, long-nosed armadillos are it. So after the fertilized egg has implanted and divided, however many times it's going to divide to produce these embryos, then you have gestation. So the female, the adult female is pregnant, and she gestates the offspring for about 3 months, close to 3 months, so that females are typically giving birth in the early spring. And after birth, the juveniles remain in their natal burrow with the mother, just nursing for about 3 weeks or so. And after that time is up, then they finally emerge from that burrow and they begin to forage on their own. And a lot of times, juveniles are active earlier in the day than the adults are. And so this is not uncommon to see what you see in this photograph, where you have the juveniles out foraging and the mother, no other adult is anywhere nearby. So they're just kind of on their own. But that's the basic sequence of what happens. So given that that's what happens, now I want to turn for the rest of the webinar to questions about what's going on with this, why it's going on, how it's going on. So there are 3 main questions that I want to pose here. And the first one has to do with the phenology that I just went through. How is it that when you have long-nosed armadillos that occupy a spatial distribution that extends from southern South America, cursor, so from this area down here all the way up to here, how can you manage to maintain that same phenology over space? As I mentioned earlier, in the United States, the armadillos here are breeding in June, but if you're down here in Brazil, presumably they're breeding now or maybe even earlier back in December. So how is it that animals are able to maintain the same sequence of events over this vast spatial distance? Well, there are a couple ideas. The bottom line is that we don't know, but there are a couple of possibilities. Generally speaking, there are 2 main ones. One would be that they're timing reproduction based on various environmental cues. There are a lot of possibilities here. One would be photoperiod, although with armadillos, with long-nosed armadillos anyway, I should say, that's a little bit doubtful because A, they're nocturnal, and B, their visual systems are not that good. So whether they could detect changes in photoperiod is debatable. Maybe they could do it, but maybe they can't. I suspect that maybe they can't. The other environmental cues would be ones that kind of go together. So they're not really independent kinds of influences. But one could be temperature, that females become reproductive when temperatures get warmer. I guess that's a possibility. Related to that would be increased prey availability, since they are eating insects primarily. As temperatures go up, you would think that there would be more insects to eat, so that would provide more resources for reproduction. And lastly, kind of again related to the same idea, would be rainfall, that when temperatures go up, you get more rain, that leads to more insect abundance. And so these 3 things together, or in some kind of interactive way, could potentially be promoting the ability to change the timing of reproduction as you move across space, so that you're still reproducing at the same ecological time of year, even if it's not the same calendar time of year, if you want to think about it that way. These are all possibilities, and certainly for those of you that work with zoo animals, I think probably not a big surprise that the timing of reproduction can be fairly plastic. You know, there are lots of instances where animals from Australia, Africa, South America have been kept in zoos or other captive conditions in the north, and they've adjusted to those new conditions and maintained their same reproductive phenology. So that would suggest that environmental cues or some kind of environmental trigger is important in controlling the timing of reproduction. Alternatively, though, just to be complete, it's possible there could be genetic effects. I just talked earlier that, Dasypus novum sanctus is not just one species anymore. There are 4 of them. And so it's possible that there are just species differences that drive when they reproduce. And so that's something that could be looked into. Nobody has done that yet, but it's possible that that could be an influence. And kind of related to that would be if there are species differences that are based on perhaps genomic differences between these species, that could be then manifested in the altered timing of gene expression and suppression in the genes that influence reproductive biology. So, and as I was saying earlier, there's no need for these things to be considered mutually exclusive. It could be a combination of not just various environmental cues, but probably some of these genetic influences as well. So that's one thing to consider with regard to how we understand reproduction in long-nosed armadillos is just how do they maintain this phenology over space. The second question that I want to address is the delayed implantation question, or the diapause. And the first part of that I wanted to bring up is, well, why wait in the first place? What prevents long-nosed armadillos from mating at some other time of year so that they could just implant the egg immediately and not wait until some later time to begin embryonic development? Well, as a piece of evidence to consider with regard to that, it's not clear how rigid or how mandatory this period of diapause actually is. Colleen, in particular, has data showing that nulliparous females, typically young yearling females that have never reproduced before, don't typically breed and mate until later in the either the late summer or early fall. But to the extent that we know, and more data would certainly help, but to the extent that we know, it seems like once they do mate, then everything else that happens on the same schedule as in older females that have mated previously. So what that suggests is that the period of diapause is plastic, that it's variable, and there's no fixed time or fixed amount of time that has to go by before females can start embryonic development. Apparently in these yearling females, they can start very quickly after fertilization, but for some reason, adult females wait. So what is it that is driving this delay between when females mate and when they begin embryonic development? I don't have an answer to that, but to extend that point, we can ask a related question, which is if diapause can vary, so it doesn't have to be a fixed amount of time, what's the cue that tells females time's up and embryonic development can proceed? Well, here again, don't have an answer. There are some ideas. Certainly be something to be interesting to try and study. But it's really the same usual suspects that I mentioned with regard to the control of reproductive phenology. It could be that there are the same kinds of environmental cues that determine when diapause ends, either photoperiod, temperature, rainfall, or prey availability, or also could be the genetic effects that I mentioned before as well, that there are just species differences, perhaps related to the timing of gene expression and suppression. But there has to be something that tells females or signals the physiology of females that diapause is over and embryonic development can commence. But we don't know what it is. And so we don't know what controls the length of this delayed implantation. Why do they need it in the first place? How long does it have to last? These are open questions. We know that it happens, but we don't know why it happens, and we don't know what controls how it happens either. So I'm just going to leave that as kind of an open issue because I don't have any data on these points. These are just, I think, to me anyway, interesting questions that we need to think about with regard to reproduction in long-nosed armadillos. And so to turn to the final question that I want to talk about, that would be polyembryony, where I actually do have some data to share with you about that. So polyembryony is, as I mentioned earlier, this is the production of genetically identical littermates from a single fertilized egg, which is extremely unique. And one question to ask about polyembryony is that there are multiple species of long-nosed armadillos. Are they all really polyembryonic? Well, I told you earlier, there are additional species of long-nosed armadillos that we didn't recognize before. In total, there are 11 as of today. Who knows what happens next week? But you can see that there is variation in how many offspring they produce. The reason for the color coding, these three species used to be considered just a single species of Dasypus capillari. Not surprising, since they were considered one species at one time. They've all got the same litter size of 2. These are all the species that were formerly considered Dasypus nubum cinctus. So again, not surprising, they all have the same litter size. But then you can see there are other species that have variable litter sizes, especially most conspicuously with Dasypus nubum, not nubum cinctus, sorry, septum cinctus, which now has subsumed as a subspecies what used to be called Dasypus hybridus, but the number of littermates that are produced there is somewhere between 4 and 12. But as I'll talk about in just a second here, it's only in Dasypus mexicanus that we've actually shown definitively that the littermates are genetically identical to one another, so that this species is clearly polyembryonic. I think the odds are very good that all of these other species are as well, but we've never actually tested it to be sure. So that would be something that would be worth doing. But getting back to the fact that these species do have different numbers of offspring that they produce, another open question that we don't have an answer for is, why is there this variation in litter size? You know, what in particular has promoted this really big increase in litter size in Dasypus septum cinctus, specifically the hybridus subspecies, where you can get up to 12 offspring, which is a phenomenal number compared to any of these other long-nosed armadillos. So is there some explanation, some reason for why litter size changes across these various groups? So as I said, I don't have an answer for that. I'll just leave that hanging. But as a second question, the one that we actually began our studies of armadillos trying to answer was whether there were consequences to polyembryonic reproduction. So you're producing these genetically identical littermates. Does that matter? And to the extent that it might matter, historically at least, the main issue, the main thing that people were interested in was how that might relate to altruism and kin selection in long-nosed armadillos. Altruism, if you don't know, just refers to a case where an individual incurs a cost to its fitness to increase the fitness of another individual. And that doesn't seem to make a lot of sense evolutionarily, because your evolutionary success is based on how many offspring, or how many of your genes, I should say, you can pass into the next generation. And so if you're incurring a cost to that, that would seem maladaptive. Why would you do that to help somebody else increase their fitness? That doesn't seem to make a lot of sense. Well, as a lot of you probably know, the answer to that came from William D. Hamilton, who developed something called Hamilton's rule, which was that the evolution of altruism is governed by this formula you see here. R times B has to be greater than C. Well, the B and C terms are kind of intuitive, that for anything to evolve, the benefit of the trait has to outweigh the cost. So in this particular case with altruism, B is the benefit to the recipient of the altruism in terms of its increase in fitness. So how much do you increase the fitness of this other individual? C is the cost to the altruist of providing the altruism, of increasing the fitness of this other individual, I should say. How much does its fitness get decreased as a result of doing that? But what Hamilton realized, really remarkable insight, was that there are two ways to be evolutionarily successful. As I said, your fitness is determined by how many copies of your genes you can get into the next generation. Well, you can do that in one of two ways. The obvious way is to reproduce yourself, because your offspring, by definition, the reason they're your offspring is that they contain your genes. But you have genetic relatives that share certain genes in common with you as well. And so you can still be evolutionarily successful if you help your genetic relatives to reproduce, even if you don't reproduce as much yourself. And that's what the R term refers to. R is the coefficient of genetic relatedness between the two individuals, between the altruist and the recipient. And so this is just a standard pedigree showing you various coefficients of relatedness, I should say, between yourself and other individuals. So you're related to your parents by 50%. You share 50% of your genes in common with each of your parents. You also share 50% of your genes in common with your own offspring, as well as your siblings, and so on. Now, in addition to genetic relatedness, though, there is one other factor that you've got to take into account, because as you can see, if you're looking closely at this pedigree, there are certain individuals that share the same genetic relatedness with you. For example, your grandparents are related to you by 25%, and so are your nieces and nephews. But if you think about it in terms of evolutionary questions, if you had a chance to be altruistic to your 85-year-old grandmother versus your 19-year-old nephew, who would you choose? Well, genetically, you're related to both of them by exactly the same amounts. So presumably, you wouldn't be able to decide. It would be the same. But the other factor besides genetic relatedness is something called reproductive value. So you would expect that there would be more kin-selected altruism between individuals that are more closely related to one another. You should, for example, be more willing to aid a sibling versus a cousin, because you're related to a sibling by 50%, a cousin only by 12.5%. But getting back to the question I posed about helping a grandparent versus a nephew, you've also got to factor in reproductive value, which is the expected future reproductive success of the individual. So what happens with reproductive value, this is just a hypothetical curve, but At birth, there's a chance that the individual might not survive until it reproduces, but as it gets closer to the age of first reproduction, its reproductive value is going up. But then after it reproduces for the first time, there are going to be progressively less reproductive attempts available in the future. And so reproductive value declines with age. And so if you're talking about aiding a 70-year-old grandmother, her reproductive value is pretty close to zero, whereas a 17-year-old nephew would be right around the age of first reproduction. So if you had a choice between being altruistic to a nephew versus a grandmother, evolutionarily speaking, just focusing on that, no other moral issues to deal with, you would help the nephew because he has a higher reproductive value than the grandmother. So that's the background. So the point of bringing all this up is that if you do expect more kin-selected altruism with higher values of R, you can't get any higher than armadillo littermates because they're genetically clones. They are identical to one another. So if that's the case, you would expect that they ought to be model systems for studying kin selection. And in fact, as I'm sure a lot of you people know, this is something that has been well known for years and was first articulated by Richard Dawkins in his famous book, The Selfish Gene, which is hard to believe. It's nearly 50 years old. I can't believe that. But as he said, you would expect a lot of strong altruism given that you have these identical quadruplets, and somebody ought to go find out what's going on. Well, Dawkins wrote a second edition to this book in 1989. And at that point, nobody had done anything with regard to studying kin selection in armadillos. And this is where Colleen and I came in, because this is what motivated us to study armadillos in the first place, was to try and figure out whether there was any kin selection going on in armadillos. We didn't start doing that until about 1990 or so. So after Dawkins, he never wrote a third edition, so he's never updated this. But what I can tell you, even though Dawkins never wrote it down, is that there is no kin selection going on in long-nosed armadillos, at least to the extent that we've been able to look. In every way that we've tried to do it, we couldn't find any strong evidence that there's any kin selection going on. So why do I say that? Basically, three lines of evidence. The first is that in order for kin selection to occur, you have to have kin available to interact with in a population. And in Mexican long-nosed armadillos, at least, don't know about the other species, but at least in Mexican long-nosed armadillos, it doesn't seem to be the case that most animals have kin available in the population. So how do we know that? This is a pretty old study, but what we did at a study site in North Florida was to screen about 300 animals for various microsatellite loci. And it's kind of hard to see here, but where it says 1, 2, 3, and 4, these are 4 different litters, but they're 4 different litters of 4 individual armadillos. So this, I don't know if you can see it very well, but this is one lane for the first litter mate in this litter. That's the second one, that's the third one, that's the fourth one. Same thing here, this is 1, 2, 3, 4. The point is that if you look within each litter, all of these bands represent various microsatellite alleles. And they're all exactly the same. They all share all of the bands completely. So there are differences. For example, you see this band isn't present in this litter, but it's present in this one. But all 4 of these litter mates do share that band. So this is genetic confirmation that Mexican long nosed armadillos are indeed polyembryonic. And getting back to what I said earlier about whether the other species are polyembryonic, we think so, but we need to do something like this where we get DNA from the various litter mates to just confirm that they are genetically identical to one another. But the other part to this is that if you sample the adults in the population, you can see that a random sample of adults, they share some bands in common, but there are others that they don't. And so the really nice thing about this, as far as we were concerned, is that, you know, when you start a study of a population of armadillos and you're catching all these adults, you have no idea whether they are litter mates or not. So how are you ever going to figure that out? Well, the answer came from studying these litters, that if you got a perfect match at all of these different microsatellite alleles, the only way that that could happen would be if they were from the same litter, because random adults don't share all of their alleles in common. So based on that, you know, we looked at the adult part of the population, and what we found is that there are very few adults that do have a perfect match with anybody else in the population. So very few adults have any litter mates still present in the population. So whether they dispersed, whether they died, we don't know, but they're not there. And not only are they not there, but in the very few cases where you actually had an adult that did have a relative present, they were widely scattered from one another. So this is another figure from the study of the same population. These black triangles represent adults that had a litter mate present. And notice, we sampled 200 some adults, and we only found 8 cases where there was a litter mate present. So there's one here, second one here, third one down here, and so on. But when you look at where they're located, you know, these two adults are so far away from one another, there's no way that they would probably ever interact. And that was true in general for the adults, that even if they did have a litter mate present, which was very rare, they were not very close to one another. So any potential for kin-selected altruism didn't seem to be very plausible. Now, the circles represent juveniles. So these would be the young that have just emerged from their natal burrows. They're foraging together during that first summer above ground. And you can see that they're very tightly clustered. So we extended what we were doing a little further and said, well, okay, maybe there's no kin selection going on among these adult armadillos, but these juveniles are certainly close enough together, they could interact, and maybe there could be some kin-selected altruism going on there. So to test that a little further, we did 2 things. And what we found out, the upshot is that we didn't find any evidence that juveniles could discriminate between kin and non-kin behaviorally. So we actually did 2 experiments. The first one was a scent experiment, where we caught very labor-intensive study, but we caught a bunch of juveniles. We kept them in plastic tubs with these little cotton pads, so the pads would get covered in their scent, you know, urine, feces, and so on. And then we would take a juvenile, stick it right down here in the middle of this testing arena with one pad here, one pad here, and we did a couple of different experiments, but the main one that we were most interested in was if one of these pads was from a litter mate and the other one was from a non-litter mate, could they tell the difference? Could they discriminate? And the answer was yes, they could. They actually spent more time near the pad of their litter mate than a non-litter mate. So they could tell the difference. But for kin selection to be important, they have to act on that difference. If you know that this person is your relative and this person is not, you need to treat your relative differently than you do your non-relative. So we did another experiment where we built this larger testing arena, and we put 2 juveniles in the arena at the same time. and filmed their interactions with one another, thinking that, well, litter mates would be very amicable to one another because they are clones, and they would be hostile to non-litter mates. And what we found was that they didn't care. You know, based on the body size difference, it's pretty obvious that these are not litter mates. The small one is from one litter, the larger one's from a different litter. And yet all of the juveniles that we tested, whether they were litter mates or non-litter mates, they all interacted amicably almost all of the time, so that we could find no evidence that even though they can tell the difference between kin and non-kin, they don't act on that difference. So we couldn't find any evidence that there's any obvious kin selection going on in 9-banded armadillos. Now, to just extend that point a little bit further, quickly, because I know I don't want to take up your whole evening here. But as an additional consideration, just kind of further driving home why there might not be kin selection going on in long-nosed armadillos, there are two people who provided some ideas that we found useful. First one was by, I believe his first name is Barry Lovegrove, who pointed out that there's a certain style of locomotion called plantigrade locomotion, which is the heel toe, so like that, where vertebrates that have or exhibit plantigrade locomotion typically also possess armor. And the armor provides protection against predators, so they don't have to be very fast. So since they don't have to be very fast, that allows them to have low metabolic rates, and Xenarthrans in general, and certainly the long-nosed armadillos are clear examples of that, where they have metabolic rates that are much lower than expected for a mammal of their body size. And because they have low metabolic rates, they can eat low-quality prey. And a lot of the insects that long-nosed armadillos eat are not energetically very rich. So it all seems to go together, that armadillos, because of the fact that they have plantigrade locomotion and the possession of armor, have these other qualities as well. Well, if you then tie that with another hypothesis proposed by an anthropologist, Robin Dunbar, he pointed out that any social interaction is costly. You have to spend time and energy engaging in social interactions. And that takes away from the time and energy that you could expend doing something else. Well, in the case of armadillos are not active for very long periods of time. They're active typically in the evening, at night, for a few hours, but they spend the bulk of their day, or the bulk of their time, underground, sleeping in their burrows. So what we thought might be going on is that, given the fact that armadillos have this low metabolic rate, limited active period, and they're eating these low-quality insects that are typically widely scattered, maybe armadillos, long-nosed armadillos, more specifically, are time and energy limited, in which case they wouldn't have the time to be spending on kin-selected social interactions. So is there any data to support that? Well, I had a master's student named Keir Ancona who did a time budget study of Mexican long-nosed armadillos, although at the time she thought they were 9-banded armadillos, naive her. And this is what she found. In case you're wondering, instantaneous samples refer to just recording what an animal's doing at the moment that you first see it. Focal animal samples involve recording everything that an animal does for an extended period of time until it moves out of sight. And regardless of which data set you use, armadillos spend almost all of their time feeding, somewhere between 80 and 90%. And certainly anybody who's ever seen a long-nosed armadillo, if you think about what it was doing when you saw it, it had its nose in the ground feeding. That's what they do more than anything else. So we interpreted this as perhaps support for the Dunbar-Lovegrove hypotheses that perhaps because of plantigrade locomotion, possession of armor, low metabolic rates, all of that stuff, maybe armadillos can't afford to spend time on complex kin-selected social behavior. Now, that's our hypothesis. Whether it's ultimately going to be the explanation for what's going on in long-nosed armadillos, we shall see. But currently, this is where we have come down on the question of kin selection and altruism in long-nosed armadillos, is that they don't seem to do it, and this might be one reason why. Now, to wrap this up then, that leaves one last question, at least as far as we're concerned, which is, well, then why are they polyembryonic? What's the point? If there's no kin selection, no altruism, why are you bothering with this? And what our personal favorite hypothesis is at the moment is that we think polyembryony evolved to increase maternal fitness. So if hypothetically you're a long-nosed armadillo female and you only produce one egg that can be fertilized, if you wanted to increase your fitness, what could you do? Well, what most sexually reproducing species do is to produce more eggs so that you can have more eggs be fertilized. But as I'll talk about in a second here, maybe there's something that prevents long-nosed armadillo females from being able to do that. Well, if that's the case, then another way to increase your fitness, that is produce more offspring that carry your genes, would be to have the single fertilized egg that you have produced split into multiple copies so that you produce additional offspring. And so as far as we can tell, or at least what our current thinking is that polyembryony probably doesn't have much of a connection with kin selection and altruism, but maybe it evolved so that females could produce more young. But that still leaves a related question, which is, well, why do this by polyembryony? The conventional wisdom with sexual reproduction is that it generates genetic variability so that you might be able to produce some offspring that would be able to survive in a variable environment. But with polyembryony, you're producing just multiple copies of the same thing. So if there is a variable environment, if one of them can't survive, none of them can survive. So how is this doing you any good? So the last thing to consider here would be, as I was starting to say a little bit earlier, maybe there's something that limits females to the production of a single fertilized egg. And maybe that has to do with some sort of physiological or anatomical constraint in females that just limits how they can reproduce. And the earliest idea about this was proposed by Gary Galbraith back in 1985, who argued that, if you remember, this is the uterus, here are the ovaries, there's one ovary, there's the other ovary, this is the fundus. He proposed that the area in the fundus where the fertilized egg sits during delayed implantation, before it implants, he proposed that that area was small enough that additional eggs couldn't fit into that space. And so females could only produce one fertilized egg. And then if they were going to produce more young, the only way to produce more would be to do that by polyembryony. I'm no expert on this, but somebody who knows more about it than me, named Alan Enders, who's done a lot of work on reproduction and particularly the placental morphology in long-nosed armadillos. He says that there is more than enough room in the fundus for more than one fertilized egg. So I'd say the jury is out on Galbraith's hypothesis. It doesn't look good, but it's a clever idea, and it would be worth looking into it further to see just exactly whether there is enough room or not. As a second alternative, as I said a little bit earlier, maybe there's some sort of constraint on ovulation. So maybe even though there might be enough space in the fundus for more than one fertilized egg, so there's more than enough room here, maybe there's something that limits females to the production of just a single egg. I don't think anybody knows. I certainly can't think of any work that's been done to show whether females can ovulate more than once in a reproductive season or not. If they can't, I don't know if there's any data that shows that they can only ovulate one egg. But perhaps there is something physiologically about long-nosed armadillo females that limits them to ovulating just a single egg per reproductive cycle. And so if that's the case, again, if you're going to produce more offspring, the only option would be to do it via polyamory. And then lastly, another good idea, but nothing has come of this one either, was something that was proposed by somebody named Bruno Reversade, who's in Singapore. And he was interested in the possibility that there's a twinning gene that promotes polyembryony in armadillos. And Mariella knows a little bit about this. But what Reversade, I should say, wanted to do was compare the genomes of long-nosed armadillos that engage in polyembryony versus the genome of armadillos that don't. For example, some of the other species like peaches or the hairy armadillos and so on, where they typically only produce one offspring. And he thought he might be able to identify differences in the genome of the long-nosed armadillos that would indicate, that would be something that was promoting polyembryony. I've looked over the years, in fact, I looked earlier this morning, and apparently nothing has ever come of his work on that. He's done some stuff looking at human genomes and looking for associations in the human genome with monozygotic twins, but apparently nothing ever happened with the samples that he got from armadillos, or it didn't work. I have no idea. But I guess the possibility still exists that there could be some sort of twinning gene that is promoting the exhibition of polyembryony in long-nosed armadillos. So with that, I'm going to wrap things up. I know this has probably been kind of a, I guess in a way, kind of frustrating webinar, because I don't have any answers for all these questions that I've posed. And if you remember, I said there were two reasons for the title of this talk. And one concerned the fact that long-nosed armadillos are not just what we thought they used to be. The other one I will talk about now as part of my conclusion, which is that we've spent all this time talking about reproduction in long-nosed armadillos. And I think most people can agree that we have a lot of data about what happens. But what we don't know much about is why these things happen the way they do and how they are generated. And getting back to the title of my talk, for those of you that may have read our book, we actually brought up most of these questions in our book 10 years ago. So you might ask, well, why are you talking about this now? And the answer is because really, if you look back over the last 10 years, there's been almost nothing done on any of these questions. So this webinar is, my hope is anyway, that this webinar might actually incite some people to think about these questions a little bit more and maybe try to answer some of these questions that have been lingering for a very long period of time. And so with that, I will shut up. So thank you all, and I hope I hope this was interesting. That's it from me.
Yes, it was very interesting. Thank you so much, Jim. You don't have to shut up quite yet. You're probably going to get a couple of questions, but first I'm going to talk just for a few minutes about the specialist group. And as I do that presentation, the participants are encouraged to type in some questions. My name is Kenny Coogan. I'm the education coordinator for the International Union of Conservation of Nature Species Survival Commission Xenarthran Specialist Group, which is a lot of words. And what that means is we love Xenarthrans, which are these adorable animals. Our website is xenarthrans.org. We have been active for 2 years on Instagram and Facebook, and also on YouTube. And way back in September of 2022, Jim was the first professional and specialist group member to say yes, he would do our monthly webinar. And ever since Jim said that, we have had more than a dozen people also commit. So thank you, Jim. And also thank you for being our first speaker for 2024. If you go to our website, you'll see that we have species accounts for all 40 species of Xenarthrans. And I guess according to Jim's presentation tonight, I'm going to have to make 3 more species profiles pretty soon.
Pretty damn, give it a couple of months, but
Yes.
yeah, you're gonna have to get used to it.
Yeah,
It's coming.
And on those species accounts, we have descriptions, we have habitat and ecology, ranges, reproduction, conservation status, diet, things like that. And that, of course, is for the adults in the room. But in the past 2 years, we have created animated videos in Spanish, English, and Brazilian Portuguese about anteaters, armadillos, sloths, and also extinct Xenarthrans. And to accompany those videos, we have activity sheets, coloring sheets, spot the difference. And all of these activities are also available in all 3 languages. We encourage you to use them in your outreach programs. And later this year, when we have our special days, we hope you pass this information around. And right when I said that, I remembered I did not change this. So this is the dates for last year, but I have to update it for this year. International Armadillo Day, always on August 13th. World Anteater Day, always. I think it's always on the 19th. Marielle is going to have to correct me. International Sloth Day, of course, changes every year. It's the 3rd Saturday of the month. I do know that. All right, we do actually have, I think, 4 more webinars coming up, or scheduled at least for 2024. And they will be advertised very soon. But the next one is around Valentine's Day, because we love Xenarthrans. And if you love them and you care for them in a captive setting, you may want to enrich them. And we will be having 3 people from the San Diego Zoo talk about how to enrich Xenarthrans. All of our webinars are free, of course. You can learn more by going to our website or our social media pages. If you work with Xenarthrans in captivity or in the wild, and you have some photos that you would like us to, or we are allowed to use, please contact us at contact@xenarthrans.org. If you feel inspired to save Xenarthrans, you can go to our website where we have a donate button, and your donations help our conservation and our education initiatives. I hope you can see my hoodie with all of the anteater family on it. We have t-shirts of lots of different Xenarthrans. And we have the giant armadillo. We recently added a 3-banded armadillo because people requested, and we follow those orders. And Jim, if you look right here, center bottom, you can get a 9-banded armadillo that has 4 little pups.
Well, that would be only if you're in South America.
Yeah.
There's no 9-banded armadillo in the United States anymore.
Well,
the Mexican long-nosed armadillo.
yes. Well, we are yes, we are supporting all the Xenarthrans all over the Americas. You can get this as a t-shirt, a hoodie, a mug, sticker, lots of options. And our monthly webinars and our educational content is funded by our partner institutions, which is FIA, the Foundation for International Aid to Animals, and Nurtured by Nature. All right, so we got a couple of people saying stuff to you. Jim, the first one we'll take is Anderson. Jim, are you aware of any study comparing the ecology of armadillos in the southern United States and to its extreme northern distribution, like Illinois? As the species are moving to much colder areas, I am curious if they are showing any change in ecology, like a broader diet or behavior.
Nothing detailed yet. There have been a couple of studies done in Tennessee and Illinois, but it's mostly just been picking up roadkills and testing them for leprosy and things. There haven't been any detailed studies. There's been some work done in Arkansas as well, but those have been camera trap studies. So they don't provide a lot of detailed information. They give you more of a sense of density and abundance more than anything else. So the jury is still out. It wouldn't surprise me if there are some changes that are occurring, but at this point, we don't have enough data for anybody to say exactly what they might be.
Right, very good. So, Mindy, a proud supporter of our Xenarthran specialist group, asks, is polyembryony a very primitive artifact that didn't have a negative impact on armadillo survival success and just persisted? And then the next question is going to be about echidnas, which I feel like those are primitive mammals as well. But go ahead, Jim.
Well, let me take care of Mindy first here. I don't think it's terribly primitive because it's confined rather to just the single genus of long-nosed armadillos. The other species of armadillos, the other Xenarthrans, don't do this. So this would be a fairly derived trait that only evolved in the Dasypus line. And you know, as far as whether it would have a negative impact on armadillo survival success, or that didn't have a negative impact, that's hard to say. But it does seem to me like it's something that is more derived as opposed to primitive. But what drove the evolution is something that, as I mentioned in the webinar, you know, there's some ideas out there, but I don't think anybody has a compelling answer just yet.
All right, so Stuart Nickel says, they work with echidnas. In Tasmania, females have a hibernation-induced diapause, which is why this interests me. I can't think of any mammals which show diapause of more than one embryo at a time. If that is the rule and delayed implantation is necessary, then polyembryony is the only way of increasing offspring numbers. What do you think about that, Jim?
That's interesting. I didn't know that about mammals that diapause, that they only have one embryo at a time. I guess, you know, you obviously know more about it than me. I will say that long-nosed armadillos don't hibernate though. So that would not be linked with delayed implantation. But if diapause is linked with only having one embryo at a time, that's an interesting connection. And maybe we're wrong about the constraint being, or maybe I'm wrong about the constraint that drives polyembryony being female reproductive physiology in terms of how many eggs they can ovulate or how much space there is in the fundus for the egg to sit. Maybe it's actually being driven by diapause, that the fact that females, for whatever reason, have to have this period of diapause. Maybe if you can only have one fertilized egg during that diapause period, if you want to produce more offspring, then the only way to get around that, like you say, is through polyembryony. So that's an interesting idea. I have to think harder about how you would test that, but maybe diapause is what's really underlying this more than anything else.
Jim, would I be able to share with the participants who are here right now your email?
Yeah, actually, I was going to mention.
Okay.
I should have mentioned at the outset, if you were looking at my title slide,
Okay.
there's no affiliation there because I am retired. But even though I'm retired, I still have my academic email address. You know, they don't take that away when you go. So my email address is still the same as it's always been. It is all lowercase, the letter J, then my last name, Loughry, at Valdosta, that's VALDOSTA.edu. But you can certainly, if you didn't get all that spelling, just look up my last name online. Nobody has my name. So if you type Loughry, Loughry into Google, you will find me immediately.
All right, very good. For people who are attending live, I put it in the chat. And because of time, we have one more question, and then maybe Mariella wants to say something. Mindy asks, has anyone done a study of how far juveniles migrate from their original nesting area as they mature?
Only indirectly. We've done a little bit of that, but it's only been through mark recapture studies. Nobody's ever done any radio tracking or telemetry studies of juveniles to see how far they go, because it's just technically really very difficult. But from the mark recapture stuff that we've done, we have found juveniles that have moved up to 1 kilometer from where they were, from where their natal burrow originally was. So they can move quite a bit. A lot of them just disappear completely, so we don't know if they died or they just moved really far away. But there does seem to be the potential, at least, for fairly long distance movement.
All right, very good. Thank you, Jim. And Mariella, our chairperson, is here. Would you like to close out the evening, Mariella?
No, thank you so much, Jim. It was, as always, was very interesting. And I'm thrilled that Stuart Nichol is here. Thank you for joining us. I love your papers, and working with armadillos, I always look at echidnas. For some strange reason, they are similar. I mean, all these weird animals are similar, so it's kind of strange. But I always get inspired by echidna papers when looking at stuff about armadillos. So it's really funny connection. We should organize a webinar about echidna and comparing them to armadillos or something like that. That would be fun.
Sounds great to me. Before you said that, I sent Stuart a private message.
No, and thank you everybody for being here. There's a lot of people I haven't seen for a while, so I'm very happy to see you all here. And I do hope that, for example, some of you could hold a webinar at some moment. Anderson, I know you're on our waiting list. Colleen, you could talk about behavior of armadillos.
Yes.
She smiled.
She smiled. That's a good start. So yeah, Happy New Year to everybody. And I'm looking forward to seeing all of you in the next webinar, which will be certainly very interesting. We love mixing up in situ and ex situ conservation because we are convinced that both are extremely important, and we need to exchange experiences with the ex situ community. So that will be very interesting too for us. So yeah, I think that's all I can say.
All right, very good. So thank you once again, Jim. And we do have webinars booked for February, March, and April. And we are still planning the rest of the year. So thank you, everyone, and have a good night.